Okay, I'm still struggling with this. I'm trying to bring up the question answers to the assessment cues on the chapter 3 from the Kaplan book. So let's see if this time it works. Even if it works here, it would be amazing. Okay, we have this. Perfect. Okay, great. Now our answers are here. So let's see. Oh, what we wanted to do was to I like to copy paste the questions to here, and then the explanations are there. So because we need to know what the questions are, so let's just do that quickly. I hope it doesn't glitch again. So let's see. We have these right here. So let's go do this. Copy. Okay, now we're getting into this territory right here. So let's actually bring these way up. Right? What is this right here? Oh, this is our progress. So let's bring this here. Okay. So is it till here? Cell releases, yeah, communication. Okay, good. Let's copy paste them quickly. We have like this. So what is the next one? The anal canal fails. Okay, why the hell did it all okay, wait a second. Why did this happen? Yeah, I don't know what's going on with this here, but let's move on for now. Canal fails. Then we have, oh shoot, this was part of that. Forget about it. We also need to have a record of the questions that we miss, so later on we can go over them again, add it to our list, and yeah, so okay. So let's now go back to this. Great. Just the following signaling molecules coordinate uterine contractions. So we didn't know the answer for this. We answered C, but it was D. So it was prostaglandins and oxytocin, not progesterone. Prostaglandins promote cervical dilation and the onset of contractions, and oxytocin promotes uterine contractions in a positive feedback loop. Let me put this on the speech.
Oxytocin is also involved in milk letdown and helps the uterus to contract, shrink, following delivery. These factors together led to D as the right answer. By contrast, prolactin, which promotes milk production, is produced following childbirth, which eliminates A, and progesterone maintains the pregnancy and actually inhibits uterine contractions, eliminating B and C. Excerpt from MCAT Biology Review 2022 to 2023. Kaplan test
Okay, nice. So we now understand that prolactin is after birth, which is for milk production. Yes, because we want to give milk. The mother gives milk, breastfeeds basically the baby after birth, and progesterone maintains pregnancy and inhibits uterine contraction. So we have a takeaway here. Prolactin happens after birth for milk production, and progesterone why is it so slow? And progesterone progesterone, is it like that? So I wrote it correctly. Progesterone maintains pregnancy and inhibits uterine contraction. So for uterine contraction, we have prostaglandin and what's the other one? Oxytocin. Not OxyContin, which is different. You don't want to do that. That's how Heath Ledger died. Fun fact. The Joker for the Christopher Nolan Joker, the second Batman movie he made. Masterpiece, fucking masterpiece. Okay, let's not get derailed here. So prostaglandin promotes cervical dilation and the onset of contractions, and oxytocin promotes uterine contractions in a positive feedback loop. So positive in a positive feedback loop. So like the more it contracts, the more it contracts. It just keeps going on until the baby's out, I think. Which of the following associations of a primary germ layer and an adult organ is correct? We got this right, I believe. Yes. So yeah, I was a master at this. Explained it well. So endoderm, cardiac, no. This is mesoderm. This is ectoderm. This is ectoderm, and digestive tract is the endoderm. Sorry, the inside of you. So, embryonic ectoderm gives hair, nails, anal canal, and the lymph is also mesoderm. So, lens of the eye, nervous system, including adrenal medulla. Oh, adrenal cortex was with the gonads, and adrenal cortex is with mesoderm. But then we have the endoderm, epithelial linings of digestive and respiratory tracts, parts of liver, pancreas, and bladder. And adrenal medulla is with ectoderm, and adrenal cortex is with mesoderm. Then we have the third question: from which of the following layers does the notochord cord form? We answered D, but the answer was B, so mesoderm. A rod of mesodermal cells called the notochord develops along the longitudinal axis just under the dorsal layer of ectoderm. Through inductive effects from the notochord, the overlying ectoderm starts bending inward and forms a groove on the dorsal surface of the embryo. The dorsal ectoderm will eventually pinch off and develop into the spinal cord and brain, while the neural tube forms from ectoderm. The notochord itself is mesodermal. Yeah, that is very specific and kind of, man, if that's on the exam, we are fucked. Excuse my language. So we have takeaway. Noto. Noto. Meso. If you remember, we also had meso compounds in organic chemistry. That was when, oh boy, I forgot. Like, it's miles away in my head. Oh, these were meso compounds were for the optical rotation, right? Like the if it's so we have meso compounds, we have a cis and trans, or not, it was not R or, man, it's really easy to mix up these things. That's why I'm trying to remember it. Yeah. So neural tube forms from ectoderm. Neural tube from ectoderm. What else do we have? Was there an endoderm involved here? No, I don't see anything. Okay. The influence of a specific group of cells. This is induction, right? Yeah, we got it right. So the influence of its ability on the differentiation of an algorithm term induction. For example, the eyes are formed through reciprocal induction between the brain and the ectoderm. So the brain has an induction with the ectoderm. That's how our eyes are formed. Competence refers to the ability of a cell to respond to a given inducer, but not the influence of the group of organizing cells. So competence is the ability of the cell to respond to an inducer. Senescence is a term for biological aging. So senescence, biological aging. Determination may be the result of induction, but this term does not refer to the general concept of the effect of one group of cells on the differentiation of another group of cells. So the result of induction is determination, but it may be, right? But this term does not refer to the general concept of the effect of, yeah. These are really nice takeaways. So I might have to copy these, or let's do it ourselves. So we have takeaway. Competence equals ability of a cell to respond to inducers. Then we have what was the other terms? The senescence, biological aging. I know that. Determination. So determination may be a result of induction. Cool. Which of the following is likely to be found in maternal blood during pregnancy? This one. 5. We chose B, but it was C. So we chose fetal, but it was progesterone produced by placental cells. Ooh. Let's go for this one. During pregnancy, the placenta produces estrogen and progesterone to maintain the endometrium. Okay, we know estrogen establishes endometrium and progesterone to maintain it. These hormones are necessary for proper gestation of the fetus and should be measurable in maternal blood because they act on maternal organs. Makes sense. Prior to birth, now we have to do the elimination. Prior to birth, the fetus is immunologically naive and does not yet produce immunoglobulins, eliminating A. So, before birth, the fetus is immunologically naive and does not yet produce immunoglobulins. That's a key takeaway. So, takeaway. Before birth, fetus does not produce immunoglobulins. Yep. Hemoglobin, immunoglobulin. Yeah, nice. It is worth noting, though, that maternal immunoglobulins cross the placenta to enter fetal blood. So that's what I was thinking. So the mother does produce immunoglobulins, but not the fetus, right? So the immunoglobulins cross the placenta. So if you give a mother a vaccination, the antibodies, the immunoglobulins, do cross the placenta to enter fetal blood. So maternal IG, however, transfers to placenta. Crosses is the right verb. Crosses placenta to fetal blood. To enter fetal blood. Crosses placenta entering fetal blood. Nice. Okay, great. There is more takeaway for sure. So fetal hemoglobin is a large protein and thus cannot easily cross the placenta. Further, red blood cells are much too large to cross the barrier themselves. This is also interesting. So HbF and RBCs are too large to cross placenta. Interesting. To cross the barrier themselves. There is a barrier, like placenta is the barrier basically. Carbon dioxide from fetal metabolism can be found in maternal blood, but the fetal lungs are non-functional prior to birth as the fetus is suspended in amniotic fluid. Carbon dioxide is transferred across the placenta directly from the fetal bloodstream. Nice. So, again, before birth, fetal lungs are non-functional because fetus is in amniotic fluid and the CO2 goes like from the fetal bloodstream directly. So, and CO2 crosses placenta directly from fetal, I think it's veins, right? Arteries, fetal bloodstream. I'm guessing it would be arteries. So, arteries. Arterioles or arteries, yeah. A cell releases a substance that diffuses through the environment, resulting in differentiation of a nearby cell. This is an example of what type of cell communication, nearby cell, paracrine. This is too easy. So we have, where we, question 6. 6, we got it right. The question stem states that the cell releases a substance that diffuses through the environment and causes differentiation of a nearby cell because the cell is acting on the nearby cell, spreads diffusion. This is an example of paracrine signaling. Autocrine signaling is when the molecule secreted by a cell acts on the same cell. Juxtacrine is between adjacent cells. The signal doesn't spread by diffusion. Endocrine, a molecule is secreted that travels via the bloodstream to a distant target. Next question. A cancer cell is removed from a patient and cultured. The cells in this culture seem to be able to divide indefinitely with no cellular senescence. Okay, what are we talking about here? So we have a cancer cell that is removed from a patient and cultured. Cool. And we want to see what protein is likely activated in these cells that accounts for the characteristics of the no aging, basically. Might be actually telomeres at the end of the day. There you go. But yeah. Cells that are able to divide indefinitely with no senescence are not exhibiting normal cell behavior. Normally, somatic cells divide a limited number of times until the telomeres become too short to be effective protectors of genomic material. When this occurs, the cells stop dividing. However, in this case, the cells have continued to divide indefinitely. It is likely that the enzyme telomerase has been activated, which allows for synthesis of telomeres to counteract shortening during replication. So what I thought was, I said it was inhibitory, right? So I thought like telomerase, the A's is kind of cutting telomeres. So we know telomeres are at the end of the DNA. So we are shortening the DNA, right? But it's the opposite. These cells are not having any senescence, they're indefinitely growing like a cancer cell because the enzyme telomerase has been activated, which allows the for synthesis of telomeres. So what telomerase does is it synthesizes telomeres. So take away telomerase synthesizes telomeres. And there's no other explanation about like growth factor beta or sonic hedgehog or epidermal growth factor. Yep. Next question. Anencephaly. The cerebrum fails to develop. Which trimester of pregnancy? This is manifested in. So we have organogenesis. I think I answered this question correctly. 8A, yes. So first trimester, organogenesis primarily occurs during the first trimester of pregnancy. After 8 weeks of gestation, the brain is fairly developed and most of the organs have formed. The severity of anencephaly suggests a defect very early in fetal development, leading to A as the correct answer. The second and third trimesters are marked by significant growth and further development. Defects in these stages may cause structural abnormalities, but the absence of an organ is unlikely. So organogenesis is during the first trimester. After 8 weeks of gestation, the brain is fairly developed and most of the organs have formed. So key takeaway. So the takeaway is first organogenesis, first trimester, second and third trimester development of organs. So if we have an absence of an organ, then it is first trimester. But if there's a structural, functional abnormalities, so first trimester, absence of organ. For these structural like malfunction, abnormality. Next question, which one is false regarding adult stem cells? The answer is A. They retain inherent pluripotency if harvested from selected organs. So we have totipotent, which is every cell. We have pluripotent and we have multipotent. I think if it's from selected organs, it's multipotency. That's why. So embryonic stem cells are controversial because they require destruction of an embryo to harvest. Okay, that's what the controversiality is implied. So takeaway. Controversial. So embryonic stem cells are controversial because, so why are embryonic stem cells are controversial? Because it requires the embryo to die, right? Destruction of embryo. It requires destruction of embryo. Excuse my alarms. Then we have adult stem cells. They're significantly less controversial but require treatment with various transcription factors in order to increase the level of potency. So, okay. Where are we here? Of embryo to harvest, whatever that is. Then we learned that the adult adult stem cells are significantly less controversial, but so adult stem cells, right? Stem cells are, so we just write significantly less controversial but require TFs, transcription factors, to increase level of potency, to increase potency. Rejection is a concern when foreign cells are introduced into an individual. Using one's own stem cells should remove this risk. Rejection is a concern when foreign cells are introduced into an individual. Using one's own stem cells should remove this risk. Okay. It's like an organ rejection, right? Rejection equals foreign. So it occurs when, right? When foreign cells introduced to someone. Having your own stem cells, using own stem cells should remove this risk. Using own stem cells reduces the risk. adult stem cells are not naturally pluripotent unless pluripotency has been induced by strategic use of transcription factors. So using own stem cells reduces risk. So pluripotency is not retained in adult stem cells unless with TFs, transcription factors. And what transcription factors do probably is just like helping transcription, right? And transcription is from DNA to mRNA. And yeah, so then we have next question. The anal canal fails. Apparently this is apoptosis. During development program, cell death occurs in multiple locations. In order to ensure development of the correct adult structures, one of the places in which this occurs is between fingers and toes. Another is the digestive tract where a central lumen is formed. If a between fingers and toes. Okay, if apoptosis does not occur correctly in the digestive tract, an imperforate anus could result. Failure of determination or differentiation would likely result in the absence of anorectal structures altogether. Failure of neurulation would lead to the absence of a nervous system and would not be compatible with life. So, takeaway. Failure of differentiation, and I believe all of them, differentiation, determination, is absence of the structure. So failure of determination or differentiation leads to absence of organ and failure of neurulation, absence of life due to messed up nervous system, right? Absence of nervous system due to absence of nervous system. Following a myocardial infarction, we're talking about regeneration here. So let's see. After an injury, we might have answered this question correctly. No, right? 11 is B. So it's incomplete regeneration. Let's see. After an injury, healing occurs by some sort of regenerative process. In humans, some tissues, such as the liver, are capable of regenerating tissue with much the same function and structure as the original tissue. However, the heart is not capable of this sort of regeneration, often forming a fibrous scar in an area of injury. This is an example of incomplete regeneration, in which newly formed tissues are not identical in structure or function to the tissues that have been injured or lost. Let's do that again. So after you're injured, some tissues like liver can regenerate themselves, but some like the heart cannot do that. often forming a fibrous scar in an area of injury. This is an example of incomplete regeneration in which newly formed tissues are not identical in structure or function to the tissues that have been injured or lost. Okay, so we have our takeaways. Takeaway. So we have, liver has complete regeneration, and heart is incomplete regeneration. And this incomplete regeneration is tissue. New tissue is not like the old tissue, right? Is it about the structure or is it about the function or both? So structure or function. So new tissue not identical in structure or function. Cool. Then we have type 1 neurofibromatosis disorder that causes formation of tumors in multiple nervous system structures as well as the skin. While all cells carry the same mutation on chromosome 17, selective transcription, which would cause the most significant tumorigenesis in which of the following primary germ tissue layers. So we have this, right? Here, the mutation affects the skin and the nervous system, both of which are derived from ectoderm. The other germ tissue layers do not lead to skin or nervous system formation. So we have here causes formation of tumors in multiple nervous system structures as well as the skin. So here the mutation affects the skin and nervous system, both of which are derived from ectoderm. The other germ tissue layers do not lead to. That's what I chose. I answered this question correctly. Yes. Next one also. So yeah. Notochord is not actually primary germ layer tissue and thus cannot be answered. C. So for this one, this is Marula, Blastula, Gastrula. After the cell division occurs, the embryo consists of a solid ball of cell known as marula. Then a hollow center forms, is the blastula, and then these cells then begin to differentiate into 3 germ layers. The embryo is considered a gastrula. Next question, 14. We answered it incorrectly. Third trimester. Respiratory distress at birth but no long-term effects. The question states that the pregnant woman was given the drug 3 days before the baby was born. It is important to remember that organogenesis occurs during first trimester. Yes, last structure to become fully functional is the lungs, because the organs were. That's a nice fact. Because the organs were already largely formed prior to the administration of the teratogenic drug, it is likely that there was no major effect on the development of most organs as a result of exposure to the teratogen. Because lung tissues are so sensitive and because they mature so late, it is likely that the infant may have some respiratory distress at birth. So there is a key takeaway here. We got the takeaway from trimesters. So the only takeaway here would be the fact that lungs are the last organ to develop, right? to become fully functional, are the last organ to become fully functional. It's like a few days before birth, I believe. And the last one we answered it correctly. Which one was false? We answered the ductus arteriosus, right? 15C. Blood flow in the ductus arteriosus from the pulmonary artery to aorta. The direction of flow is determined by pressure differential between the right side of the heart, pulmonary circulation, and the left side of the heart, systemic circulation. Unlike in adults, the right side of the heart is at a higher pressure during prenatal life than the left side, so blood will shunt from the pulmonary circulation to the systemic circulation through both the foramen ovale and ductus arteriosus. So takeaway: unlike adult unlike adults, fetal fetal blood pressure, right? This is blood pressure differential. So higher pressure. So fetal pressure differential is greater in pulmonary circulation than systemic. Which is why the flow is from the pulmonary, which is why the flow is just from the right side to the left side of the heart. Both the foramen ovale and ductus arteriosus. Which is why the flow is from the right side to left of the heart. That is for foramen ovale. For ductus arteriosus is in the ventricles, right? From the ventricle to aorta. Cool. We got chapter 3 covered. Short break, and then we go for chapter 4. After that, we only have, I believe, do we have chapter 5 too? We have. So 4 is nervous system, 5 is endocrine, then we have 11 and 12, which is musculoskeletal and genetic. So then we're done actually with bio, which is a very good news, so that we can just do practice questions on bio, a lot of them, and we'll be good hopefully for the exam. Okay, let me just pause this here and we'll be right back.